Hello Professor, I'm Mubashir Raza, and the topic of my presentation is draft genome of the common synaptic turtle Calidra serpentina model for phenotypic plasticity in reptiles. So the turtles contain the total number of species 356, and they are divided into two main suborders. One is Cryptoderia or hidden neck turtles, and second is Pleuroderia, which contains the 93 number of species. And these are divided into the 14 number of families, and the Cryptodarian consists of 11 number of families, and Pleurodarian contain the 3 number of families. So this is side neck turtles, and this is hidden neck turtles. So the turtles show a wide diversity in the habitat, and they are living in the aquatic to terrestrial lifestyle, and they have one of the threatening turtles. Turtles are one of the threatening group of the vertebrates due to the habitat destructions, international trade, and due to the over harvest. So the habitat is their and they have issues of habitat loss, and that's why a large number of their species are highly threatened. And the climatic change is one of the major issue related to these species, and the climatic changes is a major concern, especially with TSD, mean temperature dependent sex determination, because their sex is mostly determined by this TSD. And 81 number of the species exhibit temperature dependent sex determination, and temperature dependent sex determination impacts on phenotypic plasticity, and the phenotypic plasticity in which the environmental factors alter the phenotypes. So plasticity has a great genetic basis, and a large number of the species and ancestor of the turtles shows the plasticity genetic basis plasticity, and genomic resources facilitate on evolution of phenotypic plasticity to survive in different climatic conditions. And genome of the 5 different species has been sequenced, and the contiguity and completeness of the synaptic turtle genome is similar to other reptiles, and a synaptic turtle is one of the good model for turtles. It provides the baseline information for genetics, genomics, epigenomic, and transcriptomics. And the control breeding shows the variation in TSD and causes the differences in sex ratios at worm incubation. These findings provide the foundation for genome-wide association for thermosensitivity. And the genomic studies of these reptiles, the genome is very useful for studying the ecological importance traits and population genomic variations. Snapping turtle display developmental rates and allele speed, embryonic development rate in the cool soil, and the contiguous well annotated genome is critical for the epigenomic studies for temperature and oxygen level and other abiotic factors. So result of this study is genomic assembly and completeness. So genomic assembly and completeness, the initial assembly of the synaptic turtle genome using the aluminum short read provides a genome of 2.12 GB, which is similar to the other species, and it contains the number of scaffold of 17,865, and the number of contigs it contains 23 2.3 GB, And the intermediate genome assembly uh, shows that it contains 2.31 GB genome, and the number of scaffold in the intermediate genome is 16,317, and the number of contigs it contains 94,184, and the all paths uh, plus PB, Jelly and Pelon, it contains the 2.31 GB, and it shows the number of scaffold is 16,317, and the number of contigs it contains the 93,330, and the final draft of the genome that integrated the nanopore long read size containing 2.25, and it shows the scaffold N50 for the final genome is 5.59 with the long and it contains the longest scaffold of 27.24 GB. So the number of contacts and gap dropped by half indicated the, as we can see here. So the number of the contacts and gaps dropped by half, which indicate the improvements in the contiguity of the final draft genome. Synaptic turtle display the great contiguity. Here we can see that the synaptic turtle genome display great contiguity, the most often published reptiles genome. The only exception was the Maxian box turtle, which used the 10x, which used the Illumina 10x sequencing technology, and it provides the linked read to produce 4.3 fold long scaffold N50. So yet the scaffold containing N50 was 11.4 fold longer than the box turtle contig N50, and the synaptic turtles also has half, as we see, the synaptic turtle genome has half as many contigs as 1/4 scaffold as the box turtle genomes. So this suggests that the link and long read provide complementary information, improve the genomic contiguity. So here is the genome of Clitra serpentina, and the total genome related to Bosco is 94%, and the fragment it contains the 3.2%. And here the yellow color, blue color is related to the complete single copy and complete duplicated, and fragment is show the color of the yellow and missing related to the this red color. So this shows the snapping turtle reference the genome contain the both complete 94%, this one is 94%, and fragment contain the 3.2 core vertebrate genes, and this the estimate of completeness is comparable to the completeness of other genomes, and only 2.6 of the Bosco core vertebrate gene were missing from the synaptic turtle, which is a similar level of completeness reported in other reptiles. So repetitive DNA here is shows the repetitive DNA and non-repetitive DNA. The non-repetitive DNA containing most of the part, and the repetitive DNA consisting, for example, in Clydra serpentina, it contains 36% of the repetitive DNA. And in repetitive DNA, most part is occupied by LTR, then DNA, then unclassified part. So in Caricinias pictia, it contains the 29%, and Gophorus agassizi, it contains the 43% repetitive DNA, and in these results also LTR is more than DNA, and then line, and then sign, and then unclassified parts. So individual heterozygosity shows the total 3.7 million variants are found, and a single nucleotide polymorphism, it contains the 3.2 million, and these results are more in big headed turtles containing the 4.99 million SNP, and the human contain 3.07, 3.21, and 3.2 single nucleopolymorphism, and 1448 heterozygous SNP observed in synaptic turtle falls in upper range observed in 27 million species, and the relative high number of heterozygosity in synaptic turtles suggests inbreed and population bottleneck were not common in its ancestors. Genes annotation. So gene annotation, the author annotated that 20,650 protein coding genes, which is very similar to the number of the penguin turtles and deserted turtle genomes, and the remaining 162 protein coding gene in the synaptic turtle did not display the homology to other genes. The author determined the accuracy of automated annotation by conducting the manual blast-n of cDNA sequence of 2006 gene model, and the most automated annotations were correct 97.7% and 2.3% incorrect. So comparative analysis of protein coding genes. So there is the comparative analysis of protein coding genes. So vast majority of the protein coding gene in synaptic turtles are the genes in ortho groups. So they contain the 22,735, and unassigned gene is 65. So percentage of gene in the ortholog is 99.7, while the ortholog in species is 15,511. And the number of orthologs in snapping turtles, as I tell that 15,511, which is similar to the number of orthologs in pear turtles, box turtles. But there are some species, the number of ortholog, the median number of ortholog is 515. This one 515. The median number of ortholog is 5,515 species. This number is very close to the ortho groups in the tetrapods, and the ortho groups is basically the index of the number of genes families that are conserved across the vertebrates. So in contrast with these results, the big-headed turtle deserts tortoise, Chinese soft tortoise. They contain a few ortho groups, and they contain the as in the figure 13,289, 13,384, which is the difference of 700 to 200 and 2022 genes. So this number shows that the ortho groups are few in some species, and this supports the idea that birds are known to have fewer ortho groups. So here is the phylogenetic relationship of synaptic turtles between the different species. Here the relationship among the turtles based on the protein coding genes, and the synaptic turtle is more closely related. Here there is a serpentina. So serpentina is most closely related to other species of the sea turtles, and this tree also shows that the big headed turtle, this one is big headed turtles. So a big hearted turtle are a sister group, and the Chinese soft turtle, this one, Chinese soft turtle is the most divergent turtle examined here. So the ortholog overlap amongst the species. Here is the orthogroups. So orthogroups, we show the different colors. So here the orthogroup overlap among the species suggests the gene model are incomplete in the big headed turtles, desert tortoise, the Chinese tortoise, and American alligator, and saltwater crocodiles. Functional annotation of protein coding genes. So experimental annotation of protein function at a genome wide scale is impractical for new model species like the synaptic turtles, but it is possible to annotate the protein function based on the structural domains and evolutionary homology. So for this, to capture the both conserved divergent structure and functional elements, author use the interpro scan, and GO means gene ontology terms, KIG pathways, reactomes. So this result shows that the functional annotation are 13,558 protein based on the architecture and function domains. For more complete functional annotations, the author adopted the GO, KIG, and reactomes, and showed that 12,704 genes identified one-to-one ortholog to human genes, and the author results from these method suggested the result in a large set of annotation from both protein signature and evolutionary homology. So non-coding RNA. So for non-coding RNA, T transfer RNA scan SE predicted a total number of 687 transfer RNA and 43 ribosomal RNA in the snapping turtle genomes. Knowing the hairpin and mature microRNA contain a set of 204 high confidence hairskin microRNA in the snapping turtle genome. So the summary is the contiguity of assembly is greater than most other published turtles and reptile genomes. Identified genome and genetic variants provide adaptation to climates in the snapping turtle. Gene and repeat content in the snapping turtle is very similar to other turtles. Snapping turtles serve as a tractable model to identify the synaptic genes in genome environment interactions. Thank you so much for listening. That is the end of my presentation here.